penultimate one coming up now, brain and neuropsychology. Right, so this is looking at the structure of the nervous system. Okay, so remember that we are looking at the central nervous system and the peripheral nervous system. We're also looking at the autonomic nervous system and the somatic nervous system. Right, I'm not going to spend a lot of time on this because you all know this from science. So central nervous system is the brain and the spinal cord. Right hemisphere controls left side of the body, vice versa. Peripheral nervous system obviously means around the outside, and it supports the central nervous system, so it receives the messages from them. It's that peripheral is then subdivided into autonomic and somatic. So autonomic is what acts involuntary, right? So we can't control it, and in that you have the sympathetic division and the parasympathetic division. We'll look at those in a minute. Somatic controls the voluntary movements of our muscles, right? Jumping on. So we're going on to the autonomic nervous system next. So one of the key functions of it is to create homeostasis, and that is when we are maintaining constant, well-balanced internal states. All right, so if we suddenly become embarrassed and we're blushing and we're hot and we're sweating, right, then the autonomic nervous system is working to get rid of that colour from our cheeks, the flush and whatever else. So this can't be brought under voluntary control. These things will happen whether we want them to or not. Okay, then we went on to look at fight or flight response to things. So that is this idea that if our brain detects a threat, then what do we do? So the hypothalamus identifies the threatening event, or whatever it is that is going on. Then adrenaline is released, and then we have a response from the cardiovascular system. Right now, these are all physical changes, but we also have psychological changes that are going on as well as somebody is experiencing these ideas. And so this is the point where for some people what they will do is they will run to get away from it, whereas other people will stand their ground and fight against what is threatening them. Okay, once the threat has passed, then the parasympathetic division is trying to get the body back into that homeostasis. So that we've no longer got butterflies in our stomach or, a dry mouth or whatever else. So then we went on to look at the James-Lange theory of emotion. So this was the idea of what he wanted to do was to try to explain emotions like fear and excitement, right? So they needed to know the difference or the relationship between the experience of an emotion and physiological changes to the body. Okay, so sort of fit with the fight and flight theory. Okay, so James-Lange is very simply, there's an event, there's an arousal, there's an interpretation, and there's an emotion. Okay, so you have an event that happens which causes some sort of physiological arousal. Now, do we stand and do we face up to that thing? So let's go back to this idea of blushing. So we see someone that we fancy, we blush. Now, do we then go forward and speak to them even though we're a little flushed and feel quite proud of ourselves afterwards? Or do we run away and hide and then afterwards feel embarrassed that they might have noticed us, or, upset that we didn't actually take the opportunity to speak to them? Yeah. Now, there are some people where you will see no physical changes, and therefore it would appear that there's no emotion going on there. It may just be that some people are able to control that a little better, right? So evaluation of this is that we do know that emotions do come after arousal, so that's there. It's challenged by the Cannon-Bard theory, and where Cannon-Bard says, look, we experience both things at the same time, or everything at the same time. It's not like one happens and then the other one happens. And then that is followed on with a negative, in that the James-Lange theory may just be a little bit too simple. Okay, then we went on to look at neuron structure and function. So we looked at electrical transmissions, we looked at different types of neurons, the three different ones: sensory, relay and motor. We looked at the structure of the neurons. So remember, we need to know our cell body, we need to know the axon, we need to know the myelin sheath, we need to know the nodes of Ranvier, and that they're there to help signals to jump across the gaps. And at the end we've got a terminal button, right? We drew this a lot of times, right? We looked at how neurons fire, so we looked at synapses and chemical transmission. So this idea of the release of neurotransmitters, the reuptake, and then this idea of excitatory or inhibitory effects going on. Yeah. So remember that a neuron may receive thousands of signals from other neurons within a network of neurons. Some of these will be excitatory and some inhibitory. If there are enough excitatory signals compared to inhibitory signals, then summation occurs. This will cause the neuron to fire, an electrical impulse will be created. Okay, and whilst we've talked about how some things are controllable and other things are not controllable, we can learn to calm ourselves in a difficult situation, right? That's why surgeons can perform surgery without their hand shaking. We then went on to look at Hebb's theory of learning and neuronal growth. Okay, so he wanted to show that psychology and biology can be used to explain mental processes. So he said that when we learn, it creates new connections between neurons in the brain, right? So he then talked about the plasticity of the brain, how it adapts when you get new learning, and then this produces an engram, okay? So that's like a trace that the learning leaves in your brain. And it can be made permanent if we continually practice and rehearse the things that we are learning, right? So we're going back to our multi-store model that we had really early on in memory. And then he talks about cell assemblies and neural growth. So you will get these cell assemblies so that they fire together. And the more that happens, stronger the synaptic connection, and then your neurons become more efficient, so they grow, so you manage new learning much better. Okay, the evaluation of this, the positive of it is that it is a very scientific theory, has a really good scientific basis, and it has a real world application because we can use it in education. So now we talk about education as being lifelong, not just once you leave school you never learn anything new. My mum always used to say, I've learnt as much as I need to, I don't need to learn anything new. However, a weakness of it is that learning is just reduced to a neuronal level, okay? So it is a bit of a reductionist theory. It's not taking other ideas into account for how we learn. Right, we then looked at structure and localization of function in the brain. So remember, now we looked at the 4 lobes that there are in the brain. So we've got our frontal, parietal, the occipitalis, oh, there it was, and the temporal lobe. And we've got different areas that are in there, okay, so that have different functions. So remember, one part of your brain can be damaged in an accident, but other parts of your brain will adapt and learn new patterns to take over, so that, say for example, somebody has a stroke and they lose the power of speech, it doesn't mean that their brain recovers. It means other parts of the brain take over that action, and they help the person to learn new patterns to be able to speak again. All right, so we have your motor area, which obviously controls movement, your somatosensory, which does things like sensations from our hands and our faces, and then we have our visual area, weirdly enough, which is to do with our eyes, auditory, to do with hearing, language areas, so how we're able to speak. Then we also have Broca's area. Okay, so he found this small area that controls speech production. It's obviously not working in me. If it gets damaged, then you might have problems remembering and forming words. And then as further research was done, about the same time as Broca, Wernicke's area was also found. So he found people who could speak, but they didn't necessarily understand what they were saying and what they were talking about. And so again, he found a specific area where that was located in the brain. All right, then we went on to look at Penfield's study of the interpretive cortex. Okay, so this is another named study in our specification. What Penfield's study was trying to show was the psychological responses patients gave when parts of their brains were electrically stimulated, right? So he particularly looked at epilepsy patients, where they'd only had a local anaesthetic applied to them, and then he stimulated different areas of their brain and recorded their responses. It also treated their epilepsy, and he did this for over more than 30 years and over 1,000 cases. The results were that if he applied stimulation to the visual area of the brain, then a person would talk about colours, shadows, maybe outlines of objects. Okay, however, if you went to the somatosensory area, they got tingling sensations and maybe that sense of movement. Okay, and then with experiences, he talked about things that people had experienced in their past, and they were reliving these events. They were very clear and vivid, but they could still be aware of what was going on. So every time he went to that same place, they recalled the same experience. He also looked at interpretations. So when other cells were stimulated, they would report emotions, feelings that were related to particular experiences. So maybe fear, if they felt something was moving out of their grasp and they weren't able to sort it out. So the conclusion was that Penfield found that the area of the temporal lobe that he had stimulated must have a role in strong memories of previous events, right? So he talks about the interpretive cortex, which stores information about our feelings. Now, the evaluation of this positively, it was a very precise method because he is using, proper surgical stimulation when he's looking at it. However, it is a very unusual sample. If you're only using epileptic patients, then there may be a difference in the way that their brains are functioning in any case. And in later research, he had some mixed results. So therefore, the reliability of this has to come into question. All right, we then went on to have a quick look at neuropsychology. So we talked a little bit about phrenology and feeling the bumps on your head. And then we talked about cognitive neuroscience. So this is how brain structures influence our mental processes. So we talked about how structure and function of the brain relates to our behaviour. So if you remember, we talked about the amygdala that is it's located very deep inside the temporal lobe. And it works to display our emotions and process our emotions. So whether we decide to be scared or whether we decide to love something or take pleasure from something. We've looked at examples of cognition all the way through. So memory and perception are examples of cognition. And then obviously you can use that to look at mental illnesses and see whether you can find a function of the brain which is causing a mental illness to occur. Right, we then looked very quickly at neurological damage. So we have to think about localized areas. If that area is damaged, how does the brain then use other areas to overcome that? That all links in with the effects of a stroke. Neurological damage on motor ability. Okay, these sorts of things can be relearned and move forward. And also neurological damage can change a person's behaviour positively or negatively as it would go. So then we looked at scanning techniques to identify brain function. So we looked at a CT scan so we can see inside the brain, but it's not absorbing, it's not getting absorbed into your skin or your soft tissue. It does show abnormal structures, but it does require more radiation than X-rays. It doesn't give us more information about the activity of a live brain. So that's where we would go on to use a PET scan. It's the idea that it is like a radio tracer. So we can actually follow things going on in the brain. So it's useful because it shows the brain in action and it shows localization of function. However, it is extremely expensive, so it's not available to everyone. It is quite difficult to interpret what is going on, and there are ethical issues with it because you're putting radioactive glucose into the PET scans and into a person's brain. So then we go on to Fmri scans. So they look at blood oxygen levels in the brain. So this is, it shows a lot of important information about areas of the brain that are being used at any one time. It doesn't use radiation, so it's a much safer method. And the images are really clear, so it does really help to see exactly what's going on. However, again, they are expensive. It's only useful if your whole brain is being looked at, and you do have to lie absolutely perfectly still for it to work properly, and there can be a bit of a time lag with it. Okay, then we went on to Tolving's gold memory study, which is another specified study. Okay, so remember Tolving wanted to see if episodic memories produce blood flow in different parts of the brain than thinking about semantic memories. Okay, so he only had 6 volunteers, and one of them was himself and the other one was his wife, because they were injected with a small amount of radioactive gold, right? And then they were scanned to see, using a PET scanner, to see the brain activity. They did 8 trials, 4 episodic and 4 semantic, in a random order. So this was a repeated measures design because the same participants took part in all the tasks. So they had to think about a personal experience, such as a holiday, where they'd been on as a child, and they had to think about semantic tasks, so recalling some sort of historical facts from their memory. So they'd be given a signal, they'd start thinking about it, and then they'd monitor the blood flow. The results were that there were very clear patterns, differences for 3 of the 6 participants. So there was a greater concentration of blood flow towards the back of the brain for semantic memories, and for episodic memory it was towards the front of the brain. So Tolve's study suggests that these memories are in 2 different parts of the brain, but both are still long-term memory. So remember when we looked at the multi-still model, we said it was far too simplistic just to say there's one box that is long-term memory. Okay, so Tolving is showing that it's much more than that. Yeah, and it helps with us to understand localization. Okay, if we evaluate that, the positive is it did give very scientific evidence. However, of course, there is a problem with the sample. It's very small. It's obviously restrictive. It can't be representative of the whole of society. Then another concern is that episodic and semantic memories can often be very similar. So it would appear that there is some sort of overlap. So Tolving doesn't account for that in what he is saying. And that's the end.